Hello and welcome to our webinar today. Thank you so much for joining us on a perfectly serviceable Wednesday afternoon. My name is Siddharth Roy. I'm a postdoc at Virginia Tech, and I hope you're safe and you're taking care of yourself and your loved ones, in the midst of all the suffering and even death that we are seeing. We can all be grateful for, the good luck that we all share on some level. And I think the pandemic, brings great moral and scientific clarity for so many of us, and especially environmental engineers and scientists. The fact that, what you and I do matters, our expertise matters. And in the middle of this, I'm seeing some troubling signs that I thought I'd, reflect on and then move on to our three panelists. So here's an example. Obviously, the pandemic is a great opportunity for many of us, and you're seeing thousands of preprints, just flooding the internet. But this one caught my attention yesterday, and it's a great story of, like two fantastic studies coming out from hundreds of hospitals across the world, and telling you all sorts of important information that the public craves for. For example, if you have cardiovascular disease, you're more likely to get COVID and get sick from COVID, all these sorts of things. And when people were interested in obviously asking questions so that the expertise could be used for all of us, the lead author could never provide the data. The co-authors acknowledged that they'd never seen the data. And as a consequence, there has been an erosion of public confidence in scientific guidance and expert commentary. And I've seen some of it too in my work with the Flint water crisis some years ago, examples like full professors stealing the work of Ph.D. students to get NSF, NIH funding and then cutting the student out, for example, or shoddy science being done to say water filters kill people when the control sample size was 1. And there are many examples that I'm sure all of you have seen where bad expertise and bad work done has hurt public confidence. And a big reason I think this happens is what we as academics and researchers and scientists care about. The idea that, there are all these perverse incentives that are guiding our work. We're on this hedonistic treadmill where publications and funding are paramount. And we're teaching our students that, that is what matters. Obviously, we want to, counter that and keep doing good science. And we can't solve all these problems, but what we are trying to do in our own small way is have community, and we're very grateful to the Heterodox Academy for helping us do this, where the idea is, our field enjoys a reputation for truth-seeking altruism in conducting science in the service of humanity. And the ability to have rigorous conversations and debates in good faith to pursue truth that can benefit everyone is paramount. And that's what the group tries to do. If you're interested, we have more information on how you can join us. So which brings me to our webinar. I'm here because I love learning and I know almost nothing about the virus and what's going on. So I decided to bring together some of the foremost experts in wastewater, air and aerosol science, and drinking water. And the idea is, so many of us could join in the field to learn what's going on, how true expertise is being used to advance knowledge that can be used by policymakers, other scientists, and the general public to take care of themselves. And so a bit of housekeeping on how this is going to go. I did my welcome, then reflections. Dr. Lin will go first. He'll talk about aerosolization of SARS-CoV-2, followed by Ms. Camille McCall from Michigan State, and then Dr. Rhoads. And in the end, we'll have about 15 minutes for questions and answers. And through all of it, whenever you have questions, please use the chat option to send 1 or 2 questions directly to me and not everyone else, so that I can go through them and select a few and ask those at the very end. Since this is a webinar, you cannot unmute or start your video unless we do it, so you just know the difference. And at the very end, we'll try to unmute all of you so that we can at least applaud and thank our speakers. Okay, so with that, let me introduce our first speaker, Dr. Kaisen Lin. He is a postdoctoral fellow at the University of California, Davis. Kaisen received his master's and Ph.D. in civil and environmental engineering from Virginia Tech. His Ph.D. research focused on the survival of airborne viruses in different environmental conditions and the survival of viruses on surfaces. So, Kaisen, I'm gonna stop sharing so that you can start sharing your screen.
Thank you for the introduction. So my name is Kaisen Lin. I'm a postdoc at UC Davis. Today, I will be talking about the aerosol transmission of SARS-CoV-2. My talk will be centering on the open letter that 239 scientists wrote to WHO about the airborne transmission of coronavirus. I will start with the contents in the open letter, followed by the scientific evidence that support the letter. I will also introduce the progress and the challenge that to recognize this transmission route, as well as the preventive measures to minimize the risk. In case some of you are not already familiar with this open letter, here's a brief background. So this letter is led by Dr. Maraska and Dr. Milton, signed by 239 experts around the world, and sent to the WHO in early July. In this letter, experts appealed to the medical community to recognize the potential for airborne spread of COVID-19 because there is significant potential for inhalation exposure to virus in microdroplets, also known as aerosols, at short to medium distance. And the goal is to draw people's attention and promote the use of appropriate preventative measures to mitigate the spread of COVID-19. Since the start of COVID-19 pandemic, the direct contact, indirect contact, and droplet transmission have been recognized as a major transmission route. The aerosol transmission route has been largely overlooked. In fact, many people are not clear about the difference between aerosol and the large droplets because there is a continuum of particle size. Previously, there was a cutoff at 5 micrometers to separate aerosol and large droplets. However, results from modern research have suggest this cutoff is outdated, arbitrary, and incorrect. A more appropriate distinguishing feature should be the route of exposure. Ballistic droplets, which are large particles, will follow ballistic trajectory and settle by gravity. They can cause infection when the droplet reach on the eyes, nose, or mouth of the susceptible individuals. On the other hand, inhalable aerosols are particles that are small enough to suspend in the air for minutes to hours. They will follow the airflow and float in the air before being inhaled and deposited in the respiratory system to cause infection. A recent study suggested that the cutoff between inhalable aerosol and the ballistic droplets should be around 50 to 100 micrometers, depending on the humidity in the environment. If you feel confused by the definition, I'm going to use this modeling result to further illustrate the difference. For particles larger than 100 micrometers, say 400 microns here, they clearly follow the ballistic trajectory and fall on ground within a few seconds. But for particles in the aerosol size range, as you can see, the 10 micron particles, they can linger in the air as shown in the colorful pattern for a very long time. And these aerosols are responsible for the aerosol transmission route. In order to make the airborne transmission happen, there are a few steps. First, aerosol containing the virus need to expel from an infected person. Then the virus have to maintain the infectivity in the air. Lastly, a susceptible person will have to inhale enough dose of the virus-laden aerosol to get infected. We'll examine the process step by step in the next few slides to see if airborne transmission is possible in real-life scenario. We have known for a very long time that virus can be expelled from a sick person during several events, such as speaking, coughing, sneezing, and singing. This figure shows a particle size distribution from speaking and coughing. Results show that the particle in a wide size range from few hundred nanometers to all the way to 1,000 micrometer can be released to the environment. In another study, researchers collect the exhaled breath from flu patient and quantified the concentration of influenza in samples. Influenza viral RNA was detected in the expelled particle in 30 minutes. Surprisingly, more viral genome was detected in the particle smaller than 5 micrometer than in larger particles. Another side note is that when the patient wore masks, the number of influenza virus detected in the air samples were much lower, which indicates the masks are effectively reduce the viral aerosol shedding. In respect of airborne SARS-CoV-2, there have been several studies reporting the presence of viral RNA in air samples collected from hospitals. In the first study, size-resolved aerosols samples were collected at multiple locations in the hospital in Wuhan, China. Positive results were found in samples collected in the room that doctors and nurses take off their PPE and in their offices. The viral concentration was highest in aerosol between 0.5 to 1 micrometer and 2.5 to 4 micrometer respectively. Another study collected air samples in Nebraska Biocontaminant Unit and the National Quarantine Unit. The results were shown on the right-hand side of the figure. Airborne SARS-CoV-2 was detected in bedroom, hallway, and near patients. Again, confirmed the presence of SARS-CoV-2 in the air. So far, we know that virus can be released and they remain airborne in the environment. The next question is whether or not they can maintain their infectivity in the environment before infecting susceptible persons. In the follow-up study, the authors examined the infectivity of SARS-CoV-2 virus in air samples by culturing the virus with human cell line. The red bars showing the percentage change in viral RNA before and after the incubation with on the y-axis on the right-hand side. In room 7A, so if the value is positive, then the viral RNA concentration increased, while the negative means the opposite. In room 7A and 5C, there was a significant increase in the concentration of viral RNA after incubation compared to that of before. This suggested that viruses were propagating and were able to remain viable in the air. Authors also examined the structure of virus under electron microscope and observed intact SARS-CoV-2. Together, the result demonstrated that SARS-CoV-2 are potentially infectious in the air once they are released. In a more controlled experimental setting, the infectivity of virus was studied. The results show that airborne SARS-CoV-2 can maintain its infectivity up to 16 hours, which is a very long time considering the fact that residential buildings usually have an air exchange rate of several times per hour. In other words, most of the virus would have been exchanged out of the indoor environment before they lose their viability. The last step to validate the aerosol transmission of SARS-CoV-2 is to prove that these virus-laden aerosols can be inhaled by susceptible persons and cause infection. This is hard to be confirmed by experiment due to ethical reasons. However, there are many superspreader events that have pointed to the possibility of airborne transmission routes. The most well-studied ones are probably the restaurant in Guangzhou, China, and the quiet practice in Washington. In Guangzhou restaurant, according to the surveillance video, People sat apart and did not touch common surface such as doorknobs. So indirect contact and the droplet transmission can almost be ruled out. In addition, the air expelled from the index patient almost only circulated in the area where people got infected set, which is shown in this greenish plume with a blue and red arrow on the top part of the figure. No one sat on the other side of the restaurant were sick afterward. Similarly, in the quiet practice and other events, the cases cannot be explained solely by droplet or indirect contact transmission, which suggests that aerosol transmission played an important role in the spread of COVID-19. So you may be wondering how this happened and why scientists worry about the aerosol transmission route. A patient expels virus-laden particles with different sizes. The large particles will settle by gravity, and the smaller aerosols will float in the air. When the individual B here is within the close contact with a patient, usually within 1 meter, he or she is exposed to both ballistic droplet and inhalable aerosols. So the virus can be transmitted by both droplets transmission and aerosol transmission route. As the distance increases, large droplets fall on the ground quickly and aerosol remains suspended. In the same time, these aerosols get diluted. Now, individual C at short to medium distance, who is several meters apart from A, only exposed to inhalable aerosol. In certain scenarios, the diluted aerosol might still be concentrated enough to infect person C via the airborne transmission route. Many factors can affect the amount of aerosol being inhaled by person C. Generally speaking, the longer the one stays in the same space with an infected person, or the more crowded the space is, the higher risk one is exposing to. Thanks to the open ladder and many wonderful work done by experts in this field, the aerosol transmission of SARS-CoV-2 is getting a lot of attention recently. It has been several weeks since the letter was received. WHO maintained their position that viruses are mostly spread by droplet transmission and indirect contact. The leading author of the letter says he has a mixed feeling. I'm sure this is exactly what others in the field feel as well. To be fair, when I checked the WHO's website last week, I found they added a great description on airborne transmission route with a lot of details. This change at least suggests that WHO is considering this possibility. There are several challenges that hamper the recognition of aerosol transmission routes. One of the most discussed one is the relatively low R0 value of COVID-19 compared to other airborne diseases, such as measles and tuberculosis. The medical community has been trained to consider certain disease as airborne when the R0 value is high. For example, the R0 value for measles is between 12 to 18, while the COVID-19 is only 2 to 3. The relatively low R0 made the medical community reluctant to recognize this transmission route. And there are many other challenges, but due to the time restriction, I will skip them. The last part that I would like to talk about is the preventive measures that are effective in reducing the spread of COVID-19 by aerosol transmission. We are already familiar with the measures such as keeping social distancing and washing hands to reduce the droplet and fomite transmission. To minimize the risk from aerosol transmission route, we should be focusing on the ones in the blue here. First, universal mask is a must because masks are very effective in reducing viral aerosol shedding, as I mentioned earlier, and blocking particle inhalation. We should also avoid poorly ventilated indoor environment and increase ventilation anytime possible. Poorly ventilated indoor environment help the accumulation of airborne virus because the lack of indoor-outdoor air exchange and the filtration by HVAC systems. Other measures such as upper room UV and the portable air cleaners should be also considered. To summarize my talk, I explained the difference between aerosol transmission and the droplet transmission. I also showed the result from studies about the presence and infectivity of airborne SARS-CoV-2. From this, I conclude that aerosol transmission of SARS-CoV-2 is likely. Lastly, I recommended appropriate measures that should be applied to reduce the spread of COVID-19 by aerosol transmission route. Lastly, I would like to thank experts who wrote the open letter and signed the letter. Many of them are very active on Twitter, disseminating results from ongoing works, preprints, and the peer-reviewed papers to educate the public. I also want to thank Heterodox Academy and SIP for organizing this webinar, and all the participants that joined my talk today. Thank you.
Wonderful. Thanks, Kaisen. I'm sure many of us have questions about WHO and why it took them so long to come around, but we'll get to that eventually. Thank you. Next up, we have Camille McCall. Camille is a Ph.D. candidate and soon to be doctor from Michigan State University. Her research focuses on wastewater-based epidemiology for early detection of viral disease outbreaks in communities.
Thanks, Sid, for that introduction, and thank you everyone for tuning in to this webinar. So I'm going to be talking about mitigating disease outbreaks using wastewater-based epidemiology. So I'm going to start with a little bit of a brief introduction about viruses and just how they spread in the environment. Then I'm going to talk about sort of the methodology of wastewater-based epidemiology. And then I'm going to talk about a recent study that my team and I published on a large metropolitan area in Michigan using wastewater-based epidemiology and our findings. Then I'll talk about a few studies that have looked at this method with COVID-19 and just some future considerations when we talk about wastewater surveillance. So the first thing is just viruses in the environment. There are many factors that influence the spread of diseases, in particular viral diseases. These include climate change, cultural and political norms, urbanization, globalization. And so as we think about how viruses disseminate in our environment and amongst inhabitants, this is something that, you know, we can expect viral disease epidemics and pandemics to be a cause of concern both now and in the future. And we also know that when we do have a viral disease outbreak in the community, oftentimes it's not feasible to test every person within that population, especially if you have people who do not show symptoms at all. And so given what we know about viruses, about their stability in the environment, and about their inability to replicate outside of a host, this concept of wastewater-based epidemiology is gaining a lot of attention in the field of viral disease outbreaks. So what does wastewater-based epidemiology entail? And so in a nutshell, it kind of follows this methodology of you have a population that's exposed to a virus or several viruses that you don't really know what they are yet, or you don't know that there is early signs of an outbreak. And then the people who get infected from that population will then, whether they are symptomatic, whether they don't show symptoms at all, or whether they are pre-symptomatic, will shed the virus through multiple routes depending on the type of virus that it is. And so this can be through urine or feces, saliva, skin, anything like that. Those viruses that get shed are then transported into the waste streams. So these include toilet waste, gray water, like when you take a shower or when you brush your teeth or wash your face. And these viruses that are shed through these media are then transported into the sewer systems, into the wastewater treatment plant. And so the wastewater treatment plant serves as kind of a collection point for these viruses that are shed within the community. And so that's where scientists and researchers come in and they sample the wastewater, untreated wastewater, using various sampling techniques. The one that I have shown here is electropositive filter media, which is the technique that's used in the lab that I'm working in. But there are also other different sampling methods as well. The wastewater that's sampled is then taken to the laboratory, where it's concentrated, and then viral DNA or RNA is then extracted from the sample. From there, you can take that DNA and RNA, and you can further analyze it using two common different methods. One is next generation sequencing along with metagenomics, which allows for the identification of multiple human viruses within a sample. This is an untargeted approach, which means that you don't need to know what's in the sample ahead of time in order to be able to detect those viruses. It gives you kind of a presence absence analysis, so whether the virus is there or not. It can also give you a little bit of quantification in a sense that is more relative abundant, so it's relative to the other viruses that are detected in that sample as well. The other method is a sensitive quantitative method. A common one that's used is qPCR or RT-qPCR for RNA viruses. This is a technique that's used to quantify viruses in wastewater, a select virus in wastewater. You can quantify it over time or over a geographical region, so different spaces. And this will give you an idea of if you quantify it over time, let's say with the SARS-CoV-2 virus, and you notice that there are certain peaks at given time periods, this might give you an idea as to maybe the number of people that are becoming infected is increasing because you have more people that's shedding the virus. So this methodology was used in a recent study that my team and I published. And so what we did was we applied this wastewater-based epidemiology approach in a large metropolitan area in Michigan. And the idea was to evaluate this application to see if we can identify potential signs of endemic disease and upcoming outbreaks within the service community. We use metagenomic analysis to look for a diversity of human viruses within the wastewater samples that we collected. We also classified the viruses that we detected according to how they're transmitted. Then we compared the information that our findings to health data within the service area or the study area during the time of sampling. And then we used that sensitive quantification method in order to confirm the results that we got from metagenomic analysis. So what we did here was, this is an image of a heat map, and along the horizontal axis, there are 6 different sampling dates. So those are the dates that we sampled. And then right above that, there's the sampling location. So there's 3 different sampling locations for this particular major wastewater treatment plant in this area. And so all those 3 locations are their huge sewers that collect untreated wastewater from the service community and take it to the wastewater treatment plant. So we sample all 3 of those major sewers. We sample, again, 6 sampling dates over the course of 4 months. So that's from November 2017 to February 2018. And what we found was 26 different human viral pathogens in the wastewater during the time of sampling. And this is what we could detect with the method that we use. That doesn't mean that there aren't more of them. And so what you see in the heat map is the boxes that are red means that the relative abundance of that virus group is more relative to the other viruses. And the darker shaded regions means that the relative abundance is less. The white squares means that the virus was not detected at all. And so those 26 viruses fell into several different categories, being respiratory viruses, bloodborne viruses, enteric, vector-borne, and then other viruses. Another thing that we did, again, we did the quantitative method to confirm the results that we found here. And we found that 5 of 6 of the viruses that we quantified in wastewater were also detected with this method here and detected in the metagenomic samples. And so that suggested that this, these next generation sequencing and metagenomic approaches can be useful for detecting important human pathogens in wastewater. So there's two virus groups that I'm gonna look at in a little bit more detail. There's alpha virus and beta coronavirus. So alpha viruses are a group of vector-borne viruses, mainly transmitted through mosquitoes. They were detected in 16 of 18 samples. One of the primary species detected was Eastern equine encephalitis virus. And again, the health data during the time of sampling in our study area showed that there were, in fact, triple E infections within the study area. Not only that, what was also interesting was that this followed a 2019 triple E outbreak in the United States, of which Michigan had 10 of the, I believe, 38 or 39 cases just within that 1 year. Another thing, again, another virus was beta coronavirus, was detected in our samples, detected in 8 of 18 samples. Now, the primary species was human coronavirus HKU1. And this virus in particular has symptoms related to the common cold. As we know, there are many other coronaviruses out there, not just SARS-CoV-2. And so this one has symptoms related to the common cold. It can also manifest into more severe illnesses such as pneumonia. And this virus was actually detected around the time that the first SARS outbreak in 2003 was detected. There were 2 possible novel coronavirus cases that were reported within the service community during this time of sampling. Now, this doesn't mean that it was SARS-CoV-2. In fact, it probably wasn't, since we sampled between 2017, this is 2017 and 2018. Another thing that we found was that there were many cases of influenza-like cases reported within the service community. So influenza-like cases are often composed of illnesses related to whether it's viral, bacterial, parasitic. If that illness has symptoms related to flu-like, if it has flu-like symptoms, but is not the flu, then it will often be categorized as influenza-like. Okay, so that's one of the reasons why we categorize coronaviruses as influenza-like as well. So these findings here suggest that wastewater-based surveillance, as well as the methods used, are useful for kind of surveying or monitoring diseases within the population, and then also forecasting potential outbreaks that could happen. So the next couple of slides, I'm going to talk briefly about some studies that are being done, that have been done on wastewater-based epidemiology and COVID. This study here was conducted in Australia, and what the authors did was they used wastewater surveillance. They sampled from several different wastewater treatment plants. One, they sampled before the peak of the outbreak, and then they sampled 6 more times during the peak of the outbreak, which you can see at the little dots in the graph there. On the x-axis are the weeks, and then the primary Y and primary Y and secondary Y axis are the COVID-19 cases per day per public health network, and then the total cumulative cases. And so what the authors found was out of the 6 samples during the peak of the outbreak, they found 2 to be positive for the SARS coronavirus RNA. But one thing that was interesting was they used a couple of different methods to concentrate the virus, and then also to quantify the virus in wastewater. And they found inconsistencies when it came to the methods that they used, where certain methods, certain primers and probe sets used for quantification, they were able to get positive signals, but then some they were negative. And so that kind of highlights the one big limitation of wastewater-based epidemiology. And this is not just with this study, but with studies across the board, is in our sampling protocols and how we, our laboratory methods and how we go about concentrating these viruses in such complex matrices. Another study that was done recently in Massachusetts, they looked at COVID-19 in wastewater. And what they found was they found viral concentrations in wastewater to correlate quite well with clinical cases. And they also found that wastewater was able to pick up viruses shed from people who had mild cases or asymptomatic cases. And those cases typically don't show up in the clinical data. So what you see in the image there on the first panel, the blue line is the virus concentrations in wastewater. And then the dark orange line is the cumulative cases, cumulative COVID-19 cases in the study area. And then the orange line is the daily confirmed cases. Another thing that was interesting was in the second and third panel. The second panel is influenza-like cases that I mentioned before. And then the third one is clinical testing of COVID-19. And so the authors found around where it has that second dotted line here, that's around March 24th, around the time where they picked up testing for COVID-19, they found that the influenza-like cases that were, again, not related to flu, decreased as the COVID-19 testing increased. And so the assumption there is that in the early stages of the outbreak within this state, before it gained much attention, those cases could have very well been classified as influenza-like cases instead of COVID-19 cases. So in both of the studies, the authors were able to detect early signs of the outbreak in wastewater. So they were able to detect the SARS RNA in wastewater in advance to the clinical cases being reported. And so this study here was about 4 to 10 days in advance. And I believe the other past study was about 6 days. So just some further considerations when it comes to wastewater surveillance and viral disease outbreaks. There's been a lot of advancement in this field. There's been a lot of milestones that have been achieved, but there's still a lot to be done. And so, for example, some things to consider might be the detection limit. So how many people need to be sick in order for us to detect the viral RNA or DNA in the wastewater? There's so many different factors that play into this, including the wastewater composition itself, the wastewater treatment plant or the structure of it, the size of the population and things like that. So this is a open-ended question that's really true to many viruses, not just SARS-CoV-2. And another question is, does virus presence mean outbreak? Is there a baseline concentration? And so what we know about some viruses, that they are very persistent in the environment. For example, adenovirus, which is a DNA virus that causes, it could cause respiratory symptoms, but the one that is very persistent in the environment is an intestinal virus that causes gastrointestinal upset. This virus is persistent through wastewater, through treatment plants and things like that. And so we know that just because it shows up in the environment, it doesn't mean that that's cause for concern. So is this true for many other viruses as well? And will this be true for SARS-CoV-2? So much work is still needed to see, what will be the long-term prevalence of the virus in the environment. And another important thing to mention while I'm on the subject is that SARS-CoV-2 RNA has been known to be detected in human feces, which is why we can detect it in wastewater. However, the viral particles that are shed in stool are not known to be infectious. And so wastewater does not serve as a reservoir as we know it right now for transmission, but it's a great tool for us to detect the virus within the water and within the community. Another thing is geographical differences. And so how does that play a role in wastewater surveillance? Not every country has a centralized wastewater treatment system. So for countries that don't have this, how do we implement wastewater surveillance? So how do we make this tool more global? Other things are virus stability in different sewer systems. So not every sewer system is different. What is the stability of the RNA when you have a system that treats domestic wastewater as well as stormwater, industrial water, things like that? Another thing is standardization of sampling and detection methods. This is something that we need to do. Just like the previous study, there were discrepancies between the two methods used. So should we standardize these methods, and is they even feasible, given that methods might need to be different depending on the scenario or depending on the geographical location? And then also the effectiveness of the sampling locations, whether you sample downstream, which is right at the wastewater treatment plant, or upstream, which might be in maintenance holes or manholes within certain neighborhoods. And what kind of social implications could upstream sampling have? So all of these questions, you know, are really great questions that to ask for, you know, advancing the field of wastewater surveillance and advancing this field and being able to apply it to public health settings. So with that, I'd like to thank the National Science Foundation, who funded the study that I mentioned earlier on with my team and I, the collaborations with the Detroit Water and Sewage Department and Great Lakes Water Authority. Also my advisor, Dr. Irina Zakaraki, she's also working on a wastewater surveillance study for COVID-19, and they're doing great work and having promising results with that. And also have some upcoming publications. And then my colleagues, Bridgen, Evan, and Hoyun. And so with that, I thank you.
Great. Thanks, Camille.
Yes.
We'll move on to our final speaker for the afternoon, Dr. William Rhoads. William is a research scientist at Virginia Tech and the Eawag Aquatic Research Institute in Switzerland. His research explores applied environmental microbiology and chemistry in building drinking water systems. He has led multiple bench, pilot, and field scale research projects and surveys investigating the presence and remediation of opportunistic pathogens in the aftermath of water quality emergencies, disasters, and severe outbreaks. Also a dear friend, William.
Thank you, Sid. So I guess my presentation is going to be a little bit of a change in gears because fortunately, drinking water is not a source of transmission that has been documented or that we're aware about for COVID-19. So that's a very good sign, but that doesn't mean necessarily that all of the reactions to COVID-19 don't have an impact on our drinking water quality. So I was involved in a paper led by Caitlin Proctor and Andy Welton at Purdue University that documented some of the considerations for water quality implications of extended stagnation. Because we have quite a bit of experience in the water industry where stagnation or water age can adversely impact the drinking water quality in our hot and cold water systems. So the negative impacts of water age on water quality has been, very significantly documented in our distribution water systems. And stagnation can lead to chemical, biological, and physical water quality changes, many of which, highlighted in red here, have a direct potential impact on public health. And we know that in routine settings, in building plumbing systems, water is inherently more complex due to the wide variation in building design and operation. And so under even routine scenarios, water quality in buildings is a little bit more variable and kind of hard to put your thumb on relative to the experiences in the distribution system. But any negative effects of water age in the distribution system would also be expected to a higher or even more variable degree than in the building plumbing systems. And so if we think about water age, it's nice to think about it holistically, because distribution system water age is the time that it takes for water to get from the drinking water treatment facility to individual buildings. And in many systems, this amount of time can range from days to weeks from treatment to the point of entry, or POE, of an individual building. And if we think about what's happened with COVID-19 stay-at-home orders. I'm sitting in my living room right now. Many of us are working from home. And so that may have altered the way that water is flowing through our distribution systems. And there could be areas in a distribution system, if it's a highly commercial system or industrial businesses that have reduced use or reduced occupancy, where the water age in the distribution system may become locally higher than it was prior to stay-at-home orders. And of course, with building plumbing systems, the same is true. Of course, if none of us are going to work, nobody's in there flushing the toilets and washing our hands or anything like that. And so there was a study released by Cleveland Water that documented that 20% of their accounts in their distribution system had more than 25 or 75% reduction in water use. So that means that 1 in 5 buildings has drastically decreased the water demand that's through their building system on a typical day. So it's worth kind of exploring what happens during stagnation events. And one of the kind of most obvious things is that, okay, so water age is going to increase, but what does that really mean for drinking water quality? And one thing that it means is that the water is it's sitting stagnant in the pipes. So to get, quote unquote, fresh water from the utility, it may take a long period of flushing at individual outlets or fixtures in the building to achieve, water that's representative of fresh water from the utility. So in this example, in a relatively stagnant LEED building a few years ago, it took nearly 80 minutes of flushing in order to achieve water quality that was representative of what was in the distribution system. And then the flip side of this is that during stagnation, after you stop flushing the water and you allow water to just sit stagnant at the taps, the disinfectant residual that we have in our drinking water will naturally dissipate with time. And at some taps that are particularly reactive for one of several reasons, the disinfectant residual can dissipate very quickly. So in highly stagnant buildings or buildings that have reduced occupancy, it's possible that if you are reducing the water demand in your building by 75%, that you're never really achieving the fresh water from the utility in general. And if you do, it's the disinfectant residual might be decaying rather quickly. The same is true for hot water. So in hot water systems, the main control for preventing opportunistic pathogen growth is maintaining elevated temperatures. But at individual taps, regardless of what temperature your water heater or your circulating line in your system is set to, the temperature will dissipate. And in our very well insulated modern buildings, the water can sit around for 99.9% of the time at relatively amenable temperature or relatively amenable temperatures for growth of pathogens. And so this is important to consider when you're thinking about a relatively stagnant building. But in addition to this, which is more of a dynamic effect, there can also be corrosion of your metal plumbing or plumbing components, release of metals from scales. Scales can be destabilized, or loose deposits can settle out and accumulate in pipes during stagnation or in water tanks. And there are various metals that have both health and aesthetic concerns. Two of the ones that are regulated and of prominent concern are lead and copper. And so lead occurs where you have lead pipes or leaded solder or brass. And there are many legacy sources of these in our drinking water systems today. And in addition, it's interesting to note that when you have really stagnant systems, after you go in and start flushing the water, you're more likely to see discolored water at the outlet when you're flushing. And so I've seen this time and time again, where you go in after a period of time, and you get some red water discoloration in your samples. So when that happens, that metal can or that sediment can release other metals. So the orange color here is likely to be iron. I think in this case it was certainly iron. But when iron is released to water, particularly as particulates, as this would be considered, it can come with high levels of other metals as well. So lead, for instance, can absorb to that iron. And when you see big chunks of iron come off, you often see elevated lead as well. And then the other issue with stagnation, of course, is the growth of opportunistic pathogens. So these pathogens tend to grow and be released from the biofilm. And during stagnation, the biofilm in general will change structures a little bit and become more loosely attached. And so when you resume use, just like with releasing big chunks of metals, you can release a large amount of microorganisms, including opportunistic pathogens, with the resuming water use. And there are many opportunistic pathogens to consider. In the US, we have, and actually worldwide, we've really focused on Legionella. In the US, it's the only reportable opportunistic pathogen or nationally notifiable. So we have, good information on and a lot of standards and guidelines for how to control Legionella. And we generally use those standards and guidelines to be applied to other opportunistic pathogens as well, but it's notable that Legionella is not the only one. And these opportunistic pathogens are relatively good survivors in low nutrient conditions. And that's actually what makes them so adept at growing in our drinking water environment. They have an ecological niche where they can infect amoeba host organisms and use the nutrients or the amino acids within the amoeba for their growth and survival. So it's a little bit unclear what happens during prolonged stagnation, like many buildings are experiencing on the order of months. What will happen with the nutrient dynamics? It's kind of a data point that we have never really kind of gone out to, and we don't feel quite comfortable extrapolating our current understanding to these very, very long stagnation periods. But all of these things that can happen during stagnation brings up the question about, okay, what do we do about it for building managers that have to make decisions about how to operate and reduce risk in their buildings. So it brings up this idea of recommissioning, which might be needed. And so recommissioning is simply just steps taken to reduce the risks associated with water quality issues that may have developed during prolonged stagnation or due to low building occupancy. And we'll talk a little bit about these highlighted terms or these bold terms here in a second. But in general, recommissioning can consist of simply flushing outlets to introduce fresh water, good high quality water from the distribution system, for instance. It can focus on flushing outlets to remove sediment that may have accumulated, which is similar to just flushing, but is a little bit more intensive in terms of trying to very thoroughly flush all the locations in the building, all the mechanical equipment and outlets and special devices. And it can even include shock disinfection, where you introduce a relatively high concentration of disinfectant relative to what we use in our distribution systems as a secondary residual to kind of perform an immediate disinfection of the building system. And in response to the anticipated need for recommissioning, the CDC and other organizations have released guidance for reopening buildings after prolonged stagnation. And they note that we need to consider worker safety during flushing activities, because we have to send someone in here to take care of our buildings and make sure that they're safe before we get back. So we need to make sure that we're thinking about their safety. But in general, they focus on Legionella, and they have 8 steps to, to minimize Legionella growth or minimize Legionella levels before the business or buildings reopen. And of them, really the first 3 are where I would like to focus, which includes developing a water management plan, which for those of you that are not in the water industry, this is a highly tailored document that building managers can use to help manage the safety of their water. And in many instances, these plans can take a year to 18 months or even longer to fully develop and implement, because there's just so many moving parts. So this is a little bit of a limitation in this guidance, in that, as we're responding to all these new rules in building systems like mask wearing and occupancy rules, building managers may not have the resources to fully develop a water management plan. They want to make sure you set your water heater to 140 Fahrenheit. That's a very hot temperature, so that seems pretty easy to do, although you have to worry about scalding if you go too hot. And then they recommend to flush their system. And when they say this, they basically just refer to flushing hot and cold water through all the point of use outlets. And it's kind of a general, general advice: flush your system. And it's not too prescriptive, again, because of the inherent variability in the usage and designs and operation and occupancy of buildings. You can't be overly prescriptive because there's no one set of instructions that are going to work for every single building. The APA came out with similar guidance as well, and they also recommend flushing, but the specific parameters on flushing are not very well defined, and this is an inherent limitation to really not only what recommendations can work for all buildings, but our knowledge gap in terms of what will and will not work in some circumstances. So the idea, especially for hot water, is to keep the water hot enough for long enough, and do that frequently enough to disinfect the water system, or disinfect the individual outlet or set of outlets that you're thinking about. So when you flush water and the water is above the temperature at which thermal disinfection of microorganisms happens, then there's going to be some level of disinfection. But as I pointed out, if you're not continuously using this water at the point of use, the outlet, the kitchen sink or faucet, that temperature or any disinfectant that is in the water will dissipate over time. And as it dissipates, it's going to reach a point where it's no longer inhibiting growth, and there can be a period of time after it decays or dissipates naturally that there can be regrowth of these opportunistic pathogens. And so the question is, how frequently do we need to achieve these settings in order to adequately achieve disinfection in building plumbing systems? And this is something that is very surprisingly, I guess, to many people that haven't been trained in this, is not super well defined, again, because it's so variable in terms of the volume associated at every tap, the temperature, the chlorine or chloramine that you're using in your system. There are so many individual variables that can impact this. But I did want to point out that there are certain circumstances that can actually make these conditions temporarily worse in your building, in building plumbing systems, if you're not thinking through all of these variables. So I want to quickly talk about an experiment that I did a few years ago, actually, where I had a continuously recirculating hot water system, and I was operating it at a certain temperature, and the water would be maintained at a certain temperature in the tank and pumped all the way around the recirculating loop. And then I had distal taps or distal outlets that I operated to simulate high, medium, and low use. And I just want to talk about a subset of these examples to demonstrate how in some circumstances issues can be made worse by inadequately flushing. So to do that, I'm going to demonstrate what I call a regrowth factor, and it's on a log scale. So a value of 1 here indicates that there's 10 times more Legionella pneumophila which is our kind of poster child opportunistic pathogen of interest, in the distal outlets relative to the recirculating line, or what's in the tank and in the main part of the building plumbing system. And a value of 0 here indicates that there's no change from the recirculating line. And what we saw when we operated our hot water system at an average of 123 degrees Fahrenheit or 51 degrees Celsius was that there was about 70 times more Legionella and Haemophila in that stagnant, low use that got flushed once a week distal outlet than in the recirculating line. And it's interesting because this 51 degrees Celsius is kind of the minimum recommended temperature to deliver to outlets in many US-based consensus documents that are providing information for how to prevent Legionnaire's disease and Legionella growth in buildings. So this is an instance where if you have a low use outlet, that you have unintentional selection for Legionella and Haemophila. And what we think was happening in this scenario was that the hot water was coming into the distal outlet and disinfecting many of the microorganisms that are there. But because Legionella have this ecological niche where they can survive inside their amoeba host organisms, they actually can survive that thermal shock, if you will. And then when the temperature dissipates after you stop using the water, they can regrow to very high concentrations. So it's interesting that the COVID stagnation or the COVID stay-at-home orders has really reinvigorated and highlighted many of these research gaps that have been prevalent in the industry for some time. And just to kind of highlight some of them, the first one here is how stagnant is too stagnant. So there's different schools of thought on how long a building plumbing system or an individual outlet needs to be sitting around stagnant before it's going to develop water quality problems. And the reality of the situation is that, again, it's just extremely highly variable, and there's no kind of documented available research to really explain and decide under what circumstances is a day or a week or 2 weeks adequate or inadequate. So ASHRAE 188, which is a US-based organization, has offered up some recommendations, but they're not, they're meant to be feasible in their standard for controlling Legionella, and they stipulate that any building stagnation that's longer than 2 weeks is kind of a starting point to think about doing preventative actions. So then if you start into the preventative actions, another big gap is how frequently to flush, how long to flush, and where to flush. So in many buildings, it's near impossible to go through and flush every single outlet daily or even weekly, particularly in buildings that have reduced occupancy or reduced personnel coming in for maintenance and cleaning purposes during COVID for social distancing efforts. So it's unclear about what minimum or maximum level of flushing is really needed to make sure that we're maintaining good water quality throughout our building. It's also unclear about when flushing alone is likely to reduce health risks. So there are some health risks that clearly can be removed by flushing, but as with this example of Legionella regrowth after flushing, maybe you need to take extra steps in terms of reducing your risk. And so, for example, for metals, that could be including, encouraging occupants to use or providing bottled water, or designating outlets for, consumption or cooking, that you provide filters that are certified to remove or reduce the metal contaminants that you're concerned about. But then when you think about needing to take extra steps, it's also unclear when disinfection is necessary or warranted. In many of the Legionella guidelines that are out there, really the approach to determining when disinfection is necessary is through implementing a water management program. So for buildings that can't develop one before people come back to work, they're kind of stuck in this middle ground where, you know, they don't really know when to recommend disinfection or not. So this is another issue. And finally, you know, with all these recommendations, there really are realistic recommendations to validate the interventions are lacking. There's general guidance and you know, things that can kind of give you an idea about what to do, but in terms of how to really make this assessment of your building system and without a water management program is really lacking. And so AWWA has so I'm leading an effort through AWWA, which is the American Water Works Association, to develop a guideline that's focused on informing, you know, building managers on how to make these assessments and what to do, and also to help utilities know what, you know, building managers are going to be expecting from them, as well as healthcare surrounding these issues. And we've gone through quite a process. So I've got several co-authors, and then we've also sent it out to peer reviews from you know, leaders in the field and experts in public health, drinking water quality research, utilities, and private consultants. So it's been a lot of fun I've been learning a lot from these people and herding cats, and it's been a good time. So this is forthcoming. We had hopefully our last meeting today, so we will hopefully be putting this out into the world in the next week or so, or 2 weeks. So with that, I would like to acknowledge a bunch of people that have really helped me develop my thinking on this topic, particularly stagnation and the effectiveness of flushing over time. So I've been working with all these individuals and groups really since, I guess, well, my PhD advisors for many, many years, but since February or so, when we it started to anticipate water quality issues surrounding COVID stagnation. So with that, I would like to thank you, and I guess I will stop sharing my screen and we'll go to the Q&A.
William, thanks so much. We are at 2:09, which means we'll ask one question to each of our panelists and then move on to closing. So Kaisen, the first question we have for you is, how does indoor relative humidity affect particle size and viral infectivity?
RH is essentially like the ratio, like you can think of like you have absolute humidity, that's the maximum, like 100% is that's the maximum humidity, like the moisture that you can have, and the relative humidity is just the percentage to that maximum level. So RH is very important in the particle size and the infectivity of virus. So when a person expels a particle during coughing or sneezing, the particle size gets shrunk because the human respiratory system is very saturated condition. And then when the particle get into the environment, it will get equilibrium with the relative humidity in the room. And then depending on the humidity level, the particle can shrink to a smaller or relatively larger size. And that size is important because it determines if the particle will just settle down as the larger droplets will do, or will suspend it in the air for a long time as aerosols. So that's how RH affects the size. And then relative humidity also affect the infectivity of virus, and we have been studying this for many years. We know that in the typical relative humidity level in the indoor environment, say 20 to 60%, the viability of virus usually decrease as the RH increase. So 20% at 20% RH, the virus survive better, and at 60% RH, the virus decay faster. And that's also true for the virus that deposited on the surfaces, like for the fomite transmission.
So how should we think about building ventilation as we reopen buildings for people? How should we manage RH to minimize exposure risk?
Yeah, so I would suggest to maximum the ventilation as much as possible, but sometimes increase the ventilation will affect, like at the maximum ventilation level, you cannot lower the RH as you desire to be. So it's always a balance between these two, and also the energy efficiency as well. So yeah.
Sure, thank you. The next question is for Camille, and it comes from MSU. Great. So Dr. Maston is asking, several schools and universities across the globe actually are planning to use surveillance sampling for university dorms, and what is the probability that the sampling will actually contain sufficient viral load? For example, how many students must be positive for us to be able to even detect the virus?
Yeah, that's a really good question, and thank you, Dr. Maston, for your question. I think this is really interesting, and I think this is the open door for a great study in advancing the field of wastewater surveillance. So one thing that is still an open question is that detection limit, right? Like how many people need to be sick in order to detect the virus in the environment. And that's true with many different viruses that are monitored through wastewater. There's not a set answer, like 10 people need to be sick or 20 people need to be sick, because there's so many different factors that play into us being able to correlate viral concentrations to the number of people that are infected. One of the studies that I mentioned before, which I thought was really interesting, was they did this prevalence. They estimated the prevalence of people that are sick within their wastewater catchment area, given the virus concentration that they obtained from those two positive samples, and this was done in Australia. Now, one of the things to note about the samples that they detected positive was that their samples fell below reliable quantification limits, which the authors did mention that in the study. However, given their data from their qPCR results, they were able to kind of calculate the concentration of the virus. And then they also confirmed that it was the virus using next generation sequencing and also Sanger sequencing. But what they found, they used that quantification, the concentration, to predict the number of people that may be infected. And they came up with between, I believe, about 170 to about 1,000 people, which was the upper limit. And this kind of fell, the range of people who were, the range of cases that were reported within that population fell within that. And so I think that just goes to show that, that's a significant number of people, especially if you're dealing with a dormitory size population. You don't want that many people to be sick before you can detect the virus. But another thing with this situation is that that study did downstream sampling at the wastewater treatment plant, which makes a huge difference when you're talking about upstream sampling, because a lot of the different factors, for example, the amount of time that the wastewater spends in the sewage pipe and those kind of microbial processes that take place, it's a lot shorter when you're doing upstream sampling, which means that your sample could be more representative of that smaller population. And so I think, in this case, it's really interesting that, I'm assuming that a lot of these universities will be sampling at manholes and things like that are near the dorms, or I don't know how they will go about doing sampling. But it will be somewhere within that confined area, which can make a big difference. And so they might be able to detect, maybe if 10 people are infected with the virus, that might actually be enough to show up in the wastewater. And another thing that would be interesting with this is because it's a smaller number of people, when you're talking about a dorm size, then if something shows up in the wastewater, how many, is it feasible to test, all of the students in that particular dorm to see how many people are actually infected and, and what's the concentration that you see in the virus. So I don't know the scope of the study, but I think that would be really interesting and useful information to put out there if that's something that can be done. Because again, this is a question that's true for a lot of different viruses in many different areas with students.
Right. Just a quick follow-up. Bob Halsey wants to know, of all the methods that you've seen and reviewed and worked with, which one shows the most promise in terms of increasing the concentration of in wastewater samples in count per milliliters?
Mm-hmm. Yeah. So I can speak a little bit on the method that I use in the lab that I'm working in. The method we use is the electropositive filter media. And so it's a column filter that wastewater gets passed through, and that kind of concentrates the virus on site, so to speak. That also allows us to sample a very large amount of wastewater. And so let's say if you're, doing like a composite grab sample, for example, you might take 1 liter, 2 liter. But with the method that we're able to sample 10 to 12 liters of water. And sometimes more. And so that increases the chances that we might be able to find some of these less abundant viral concentrations in the water. And so that's one method. There's also, I think those are 2 that I commonly see. Ultrafiltration is also another method that I've seen that has been useful for concentrating viruses. And yeah, a lot of it also depends on the type of virus. So they're saying with the SARS-CoV-2, since it's an envelope virus, that can make a difference. The sampling method makes a difference in whether or not you're able to detect it.
Perfect. Thank you, William. So obviously with so many different kinds of buildings across the globe, this is a hard question to answer, but I'll still give it to you. What can be done for corrosion control in potable water at the individual building level? Like what are some simple recommendations that you'd give out to a building manager, for example?
Well, I don't think that I would. It'd have to be very special circumstances where I would encourage an individual building owner or manager to implement corrosion control for their building. So corrosion control is mandated by, our drinking water regulations. So the water that is supplied to buildings has corrosion control in it. And I would try to rely on that. Now, obviously there are exceptions to this, when corrosion control is temporarily stopped. So hopefully we won't re-see one of those instances. But corrosion control can become less effective, which speaks to kind of the scale destabilization during stagnations and the big slugs of particulates that you can see after stagnation in some buildings. And I would say that using flushing to remediate that would be the the kind of line to toe. But if you have outlets that are designated for drinking, then it would be a good idea to provide filters, or at least verify that those outlets are not leaching elevated levels of metals. So you can just kind of take a sample after you've implemented some of your flushing, if you're doing flushing, using the best practices of sampling with stagnant water and all that kind of stuff, and confirm that your outlets used for drinking don't have an elevated level of metals. And if they do, then you can think about what you might need to do in terms of flushing or filters to remediate that. But I would not. It would be very rare to recommend corrosion control practices at an individual building.
But for pathogens, the opportunistic kind, there is the possibility that disinfection at the point of entry is good advice.
Yeah, so that is commonly done where there are documented issues with water use and the system operation. So it's you know, it's prevalent. I guess I wouldn't say it's common, but yes. And when you have high risk occupants as well, so it's more common at places like long-term healthcare facilities or hospitals. And it's usually in response to a very specific water quality problem that they're trying to address. I would not recommend that most buildings think about installing point of entry disinfection.
Gotcha. Thank you. Well, with that, I want to thank all our wonderful speakers, Kaisen, Camille, and William. We haven't figured out the technical way of unmuting everyone so that they can clap. So we'll all clap for ourselves, I guess. Let's do that. If people wanted to join in, this is awkward. Big thanks to Heterodox Academy for helping us do this. And we'll post a recorded version of this online if people missed anything. And please feel free to email our speakers if you have comments or more questions. Their email addresses are on the screen. And thanks so much. Stay safe and spread all the love and light you can. Bye, everyone.
Thank you.
Bye.